Thank you to Talgat, Marie and Henk for the introductions. As been introduced 3 times, it's quite nice, feel quite popular. But I'm Thomas Pottage, and I have my email address there as well. So if there are any specific questions that you think about afterwards, you can email me. As mentioned, I'm a project team leader within the Biosafety Air and Water Microbiology Group. I'm sort of an environmental microbiologist by trade, but I have a lot of experience in waste management as well, both in the laboratory but also in the healthcare setting. And as Henk mentioned, at the moment we're doing quite a few studies investigating the persistence of SARS-CoV-2 in the environment and as an aerosol, but also actually working at high containment to investigate its survival through different decontamination approaches. And we've been out into different environments, and actually got a couple of colleagues going out to another workplace this week to do environmental sampling to see if we can actually detect it, and we can use these results to generate sort of a risk-based model for different environments as well, which can be fed into the UK government. Wait a second, sorry. After an introduction from me, obviously I've just given it, I'm going to sort of go through what the current issue is with contaminated waste in different environments, and then sort of explain a bit about SARS-CoV-2 in the environment and why this matters for waste, and also why this sort of can feed into the risk assessments as well, and what's the actual current knowledge of SARS-CoV-2. Then also we're going to look towards the back end of the presentation, look at the specific waste management in different environments. So we look at healthcare, laboratories, residentials and other areas as well. And throughout I've put references at the bottom of the slide. So when you have these, you can sort of have a look at the documentation which I've taken the information from. But also at the end of the presentation, there's a couple of slides which have all the references which I've used in it, among some, and then from there you can sort of garner some more information from spreading out of those references. So what's the problem? Obviously the emergence of SARS-CoV-2 causing the COVID-19 infection has caused a pandemic. And this has actually led to an unprecedented level of PPE and RPE, so personal protective equipment and respiratory protective equipment. As a respiratory virus and respiratory agent, then obviously we need to make sure that people are protected. And obviously at the moment there's lots of public wearing masks going about their daily work and daily jobs and just daily life. So we're actually generating a lot of RPE, but there's also other PPE, so people are wearing gloves or other face coverings as well. So this in turn has led to a sort of an unprecedented level of increase in waste, and it's not always sure how this waste should be dealt with. Do we deal with it as infectious waste? Do we deal with it as non-hazardous waste? Is it offensive waste? And it's leading to some of the waste handling systems being overloaded. And it's important to identify that segregation and separation of this waste can lead to reducing the load on some of the more intensive work or waste handling streams. So just a bit of background about SARS-CoV-2. I'm sure a lot of you already know of this. It's a severe acute respiratory syndrome coronavirus 2, so it's a bit of a mouthful. It's a lipid envelope virus, which actually will play an important in a couple of slides time. It's an RNA virus, so actually when looking in the environment, we look for the RNA as well as the viable virus. It's transmitted via aerosol routes and fomites, so contaminated surfaces, and it's classified as a risk group 3 agent. And again, I'll come on to that when we're dealing with that in the laboratory. So just looking at the transmission route, it's really actually quite important, that it has quite a big bearing on the waste and that hazard from that waste. And it's transmitted in sort of 3 key areas here. And so you can see in this cartoon on the bottom right hand corner just shows how the respiratory viruses are transmitted. So we have respirable aerosols in red, and they are the smallest particles, and they're less than 5 microns, and they will actually get into the alveoli and into the lower regions of the lungs. Then we have the thoracic aerosols, which are up to 10, and they'll get into the throat and the esophagus and down into the trachea. And then we have the larger particles, which are the nasopharyngeal aerosols. Now, they're sort of aerosols, but they're not true aerosols as well, because unlike the respirable and the thoracic aerosols, which will remain in the air for quite a period of time, the larger particles, such as the nasopharyngeal aerosols, will actually deposit out onto surfaces more rapidly. And this is one of the reasons why the sort of the 2 meter rule or 6 foot rule has been put into place, because these aerosols will not transmit too far, or at the concentrations potentially that will cause infections. And as we get to the larger, more spray-borne droplet-like, so the ones in blue, let's have a, sorry, if you can see my mouse cursor here. These are more ballistic particles, so they're quite large. They can be up to 200 nanometers. And they're, as ballistic particles, they're sort of spread by coughs and sneezes. So we have the larger particles which can actually land directly onto the mucosal membranes of the person and actually cause a site of infection there. But also we have, with the larger particles and even with the smaller particles, we have what we call a deposition rate. So basically the larger particles deposit out onto surfaces a lot quicker than the smaller particles. And this can lead to contamination of surfaces. In this case, it's a mobile phone being passed from someone, but this could be door handles or tables or cutlery or something like that, which actually, when someone coughs and sneezes, large particles can land on these, and that can present an infection risk. So this is one of the reasons why PPE is taken very highly. So if we're wearing RPE, so we're wearing a mask, that can actually limit those particle spread quite a lot. So we're reducing that potential infection route to the person we're exposing. But also hand hygiene plays a very big role. So if we're washing our hands, then there's less chance of anything we are touching actually sort of coming off our hands onto our mucosal membranes. And there's obviously the risk will vary according to the distance and also the sort of environmental conditions. As we can see, if we're in the open air, then we have a lot more wind, which is going to remove those particles away from us. So then this next slide just shows sort of a topographic view of viral genome loading in an infected person in relation to their infection and the time since infection. And what we can see is in the initial stages, and actually in the pre-symptomatic phase, we get a rapid increase in viral genome within that person. And that can peak sort of up to sort of 5 days after symptom onset. And then that actually rapidly decreases as well. And what we show here is actually the level of viral genome copies within that person is actually reducing quite a lot by the time they are ill enough for severe or critical illness and actually admission into hospital or ICU, so intensive care admission. So what we can see is potentially these people who are most ill aren't actually as infectious as we thought, and it's these people earlier that can be infectious or can be more infectious. So we're just going to have a quick look at the environmental persistence of SARS now, because this, as I mentioned before, this plays quite a big part in the transmission through the environment, and sort of looking at the survival in the air. It's not a true waste issue, but it can obviously cause, it's an area of concern, because obviously we're looking at implementing RPE and PPE to people in specific environments, but also in the general public as well, if they're wearing those, if they are closer than sort of 2 meters to other individuals. But also looking and sort of knowing how things survive in the air sort of plays a big part in identifying sort of the risks from aerosol generating procedures, such in the healthcare. So we know what PPE or enhanced PPE that healthcare workers might need to wear if they're doing things like bronchoscopy or chest physiotherapy. But that's from what we found, and what other numerous other manuscripts and groups around the world have found. There's actually a limited recovery of viable virus from around patients, but also in sort of workplace environments. And this shows that I think potentially it can be a lot more ballistic transmission rather than a sort of true aerosol. A lot of these aerosol samplers are designed to sample very small particles, and maybe they aren't picking viable virus up in these small particles, and it's the larger size particles which might contain it. But that's sort of a hypothesis which is being looked at the moment. But SARS-CoV-2 RNA is being detected in more air samples, so actually we're detecting the genetic material of SARS-CoV-2 rather than the viable virus, and those detections are being undertaken by reverse transcription PCR. So we're looking at very specific sort of assays within those air samples. And the US Department of Homeland Security has actually produced a calculator which estimates the airborne decay of SARS-CoV-2. And I've just put a screen grab in here, and you can see it's from this website on the bottom left. And actually that can help to sort of estimate the time it takes for virus decay. And we can see here, down here, it's sort of using a UV index of 2 at a temperature of 70 degrees centigrade, Fahrenheit, sorry, and 40% humidity. The actual half-life of the virus in the air would be about about 10 minutes. And to get 2 log reduction will take about 66 minutes. So it's actually quite a useful calculator, and you can sort of have a play around it yourself and see what the different environmental parameters will cause for the viability. So in a bit more focus on survival of surfaces, these are probably more appropriate for waste, as this is going to be what people are going to contact more in terms of that waste management and the waste handling. SARS-CoV-2 is being shown in the Kasloff and Riddle papers to be persistent on different surfaces for up to 14 to 28 days. Now these are using quite high concentrations of the viral suspension, which are actually sort of artificially high, so they're high enough that we can study any effect over a period of time. We can sort of have a bit more clarity on those results. But they also add a lot more protein to the inoculum, so what the contaminating media, and they can show that actually with additional protein, it does increase persistence in the environment, or in a laboratory environment on surfaces. And what we've done is we've actually shown that without or reducing that protein on hydrophobic non-porous surfaces such as stainless steel, so common hand touch sites, we're actually getting recovery within or over 7 days, but actually that viability does drop off after those 7 days. And other people's work, so Buryakov has undertaken some work, and they've actually shown that survival will actually decrease with increasing temperature. So they looked at different temperatures from 20 degrees up to 40 degrees centigrade, and also increasing humidity. So at 20 degrees, they looked at humidities of 20%, relative humidity up to 80%. And they found there was statistically significant drop in viability with an increase in temperature up to the higher ranges, but also with an increase to the higher ranges of relative humidity, which is sort of what's been shown within the aerosol route as well, where higher humidities actually cause a decrease in the viability compared to lower humidities. So this is just a couple of slides or a couple of graphs from our work by Susan Payton, and what we can see here is we've actually got recoveries in orange of SARS-CoV-2. So this is viable virus we've recovered by plaque assay, and then we've also assayed the same sample for RNA, which we've assayed by reverse transcription PCR. And what we can see is the viability of the virus drops off relatively quickly, and but the RNA is actually a lot more stable. So this is really important when it comes to sort of looking at surface sampling and interpretation, that you might be detecting the RNA, but actually what does that mean in terms of viability? And actually the RNA remains relatively stable, which can sort of, in the terms of interpretation of results, can actually show, you could interpret that as the virus is remaining stable as well, but actually what we're showing is the viability of the virus is decreasing quite rapidly. So over 7 days, after 7 days, we're not recovering any virus, but we're still recovering the RNA. So that could insinuate that a surface is contaminated for a long period of time, but actually that virus is not viable. So it's actually not going to be an infectious surface. And as I mentioned, with the hydrophobic non-porous surfaces, sorry, I touched the screen. With the hydrophilic porous surfaces, we actually get a much quicker reduction or recovery in viable virus. And this is thought that actually, because on hydrophobic surfaces, the inoculum for droplets form a droplet, and they dry, and they have a protective effect from the surface area. So the viable particles within that droplet are actually protected, but actually on sort of porous surfaces and hydrophilic surfaces, they're wicked away. So they're sort of drawn through the strands. And then this exposes those particles to more environmental pressures, which can cause inactivation. So in terms of environmental contamination levels, as I mentioned, the levels used in the lab studies are actually artificially high, and they're investigated using recovery methods which are very, very efficient. And this is something which is being looked at the moment, is the transfer from a surface to a hand, and a hand to a mucosal membrane, and seeing what level of recovery that is, because obviously that's not going to be 100% efficient, so there's going to be losses in that sort of process. As I mentioned again as well, most of the surface samples are analysed by PCR, but that only detects nucleic acid or genome copy, so it doesn't actually detect viable virus. And using this PCR, I can see that most of the environmental surface samples are greater than 32 cycle threshold, so the point where the number of copies of genome copies are actually detected in the assay. And this is quite close to the detection limit of the assay. And actually what we've shown is interpreting these CT values would show that at worst case, then any viable virus on those surfaces would be inactivated within 48 hours, rather than sort of that long period of time, just because of the initial loading or contamination on those surfaces. So initially there was an uncertainty between the split between aerosol and fomite transmission for SARS-CoV-2 and the sort of COVID infection. But actually using this data, it's now much more widely thought of that actually surface transmission is less likely, and the aerosol route plays a much larger part. And actually the CDC have used a quantitative microbial risk assessment, and they calculated that that contact with each contaminated surface for a normal person has a less than 1 in 10,000 chance of causing an infection. So you can see that actually, in that case, then the transfer from surfaces is going to be quite low, and actually the aerosol or the spray-borne route is going to be much higher in terms of risk and likelihood. And obviously, as soon as we go into the outdoor environment or harsher environmental conditions, so as I mentioned, increase in temperature or increase in relative humidity, that risk is actually going to drop even further. But also things like UV and airflow as well, with the desiccation of the viable particles, or the virions will actually increase with a greater airflow. So what does this mean? Surfaces are actually generally to be unlikely to be contaminated around people with large numbers of SARS-CoV-2 virions. The virus is shown to rapidly inactivate as well, but also as a lipid envelope virus, we can actually see using the classification of disinfection they're a lot more susceptible to disinfectants and environmental pressures than other organisms. So actually they're sort of the most easy to kill out of all of the organisms, the different groups of organisms that are around. And we can use RPE to decrease surface contamination further, so we can stop that spread from an infected person. And then also obviously including hand hygiene can really help to reduce that infection risk, again from surfaces, but also from aerosol routes or sprayborne, which might contact our hands. But obviously, there are environments where waste can be contaminated and needs to be effectively dealt with to reduce this risk of infection. Cool. And now I'm going to go on to sort of the waste management side of this sort of presentation. And we're going to look at specific environments. And we need to think about what needs to be done with waste. So we need to think that actually we need to include and undertake some risk assessments. So we need to identify the hazards from waste we're actually dealing with and what's the likelihood of contamination. And this is where we can use scientific literature and guidance documents as well to make sure that we can really sort of properly risk assess any issues with waste or any foreseeable issues with waste. And then we can use this also as well to help with the separation and segregation of waste, so we can identify the different routes for different items, so we can reduce the volumes of waste going into more intensive waste handling streams. So if we look at healthcare, now, actually, what's the sort of guidance from the WHO and NHS England and the CDC and the Health and Safety Executive in the UK says that infectious medical waste produced from healthcare facilities with COVID-19 patients should be treated like any other infectious clinical waste. So if we actually have hospitals or healthcare facilities that we're dealing with other infectious patients infected with hazardous agents, then we should always have waste streams where we can actually deal with that. But obviously it's identified that there will be more waste generated due to the increase in PPE and RPE, and this needs to be sort of dealt with. And this is not just in terms of the wards where the infected patients are, but this is in the general hospital environment. But these should be able to dealt with within existing waste streams. So we'll sort of briefly touch on the waste streams that need to be looked at. So in terms of segregation, transport, storage and treatment that should already be in place and can be adhered to. So in terms of segregation, general waste should really be separated from infectious waste. Obviously, if we've got infectious waste coming from a treatment ward, then that needs to be dealt with infectious waste. But other areas within the hospital, it might be appropriate to actually segregate that waste into more sort of non-hazardous waste, so it can be dealt with in different waste streams, so we're not taking up that intensive waste management. And we need to make sure that we're actually separating the waste based on the category. Obviously, if we know we have an infectious waste, or as down here we have sharp boxes, or we actually have just domestic waste or medicinal waste, we can actually sort of separate those out and use specific containers which are specific for those wastes. So here we obviously have a sharps box, which is rigid and sealable, so there's not going to be any hazards from any sharps contacting with the person handling that box, and that box can be incinerated in our waste streams. And also we should be able to obviously always color code and label our waste, so we can actually identify the waste easily, and the people know what the risks and hazards are from that waste because of the labeling system. So if we're dealing with transport of waste, we really need to have designated routes for the waste transport. So we actually use those same routes constantly, and these can be very identified as low sort of public routes, which is also one of the reasons why I suggest having collections at designated times of the day. So we're sort of making sure that there's less people around, so there's less risk for people if there's any issue with that waste. The trolleys, again, used to transport the waste should be appropriately labelled in terms of a hazard sign and other unique identifiers. And we should make sure we separate the infectious waste so it's not transported with any other types of waste. And really, these trolleys should be lockable and appropriately sized so they're easy to move, and they're not too restrictive in terms of sort of a movement and just general movement, so they can be easily moved. And in terms of offsite, if there's no national guidance for off-site transport, then the guidance and UN recommendations for the transport of dangerous goods has some model regulations in there, and this can be found at this web link at the bottom there, and it's available in a number of different languages. So if we're storing waste, we should limit the time we're storing it for as much as possible, and ideally we should be taking it directly to the waste treatment centre as quick as possible, so we're actually inactivating that waste as soon as possible. Storage times, though, obviously it's not always possible to actually inactivate that waste straight away or transport it. So what we sort of recommendations from WHO are that actually if you live in a temperate climate, then in winter you can store that waste for 72 hours and in summer for up to 48 hours. But in warmer climates, in the cool season, you'd actually store that waste for 48 hours, and in the warmer seasons it should be only stored for up to 24 hours. This waste storage areas should have floors and walls which should be sealed or tiled, and this basically will help to prevent any leakage of waste which might occur, but also it means that they can be easily disinfected as well if there is a waste problem or waste leakage. We want to try and restrict the access as much as possible to that waste storage environment, so we have managed access, so we have lock or key code on the door, and we also obviously have correct labelling. And this might be labelling internally into the storage area as well. So certain waste will be stored in different areas within that storage area. So waste inactivation, obviously ideally it should be undertaken on site prior to then safe terminal disposal. So if you are autoclaving waste, then that will reduce that waste hazard, and then the waste coming from the autoclave can be dealt with either as non-hazardous waste, so it can either go to municipal waste or incinerated afterwards. Now, the best methodologies for waste inactivation is high temperature, and obviously high temperature and steam, or through autoclaving or through hydroclaving, where the water within the waste is actually used to steam it, or incineration as well. And obviously we need to think about environmental conditions there, and if we are incinerating in terms of emissions. And we need to ensure that our workers who are handling the waste have appropriate PPE. So we need to make sure they have gloves, and it might be the case that need to be puncture resistant gloves if they're likely to be handling sharps. Need a long sleeve gown. We need RPE, as well as face coverings or face shield. And then also rubber boots. So these again need to be thinking about if they are puncture resistant assault as well, if we're handling with sharp instruments. But as an interim measure, safely burying healthcare waste may be done if it's unsustainable to actually use these high intensive waste management options. So it's obviously identified that some healthcare facilities will have a waste contractor employed, and we need to make sure that external waste contractor is licensed, and we have regular inspections within that licensing agreement to make sure that you are happy that that process is being is fully understood, that they're using, and also that it's calibrated and producing the effective inactivation. So this might be through the use of biological indicators or chemical indicators, depending on the process. Okay, so Moving on to laboratory setting. So let's have a sip of water. Now we have a couple of different types of laboratories. So we have diagnostic laboratories, and within these, we're likely to see through the diagnostic assays, we'd like to see lower volumes of samples coming, say 1 or 2 mils of maybe sputum or blood, and they're likely to have lower concentrations of the infectious agent in it. But also, as I mentioned before, they will be quite highly, have a high level of protein or organic material in them, which actually can lead to a greater viability over a course of time within those samples because of the protective effects of that contaminating media. But also we're likely to see other pathogens in the sample, so it's not just SARS-CoV-2. If someone's coming in presenting with respiratory issues and they test negative for SARS-CoV-2, then it's potentially going to be another respiratory pathogen which they have. And there's obviously other agents out there such as TB, which are obviously hazard group or risk group 3 agents as well, which we need to be wary about and protect our staff. But there are likely to be lots of samples, so it's likely to be these laboratories will be sort of the focal point for lots of samples coming in. So there's likely to be lots of samples, so it sort of offsets the low volumes because we will have lots of samples. Now, if we're looking at a research laboratory where we're actually propagating the virus, it's likely only to be SARS-CoV-2 because we're only growing that virus and we're using that for studies, or we might be using that for assay validation or disinfection validation as well. And we want to make sure that we are handling that safely because they're likely to be high volumes of high concentrations. So there's likely to be obviously a bigger risk there if there is a spillage or leakage or non-inactivated waste leaving the laboratory, because it's going to be at like high concentrations and it's going to pose a greater risk. But we're probably likely to have a smaller number of samples than the diagnostic throughput laboratory. As I mentioned previously, SARS-CoV-2 is a risk group 3 agent, so obviously we need to handle that appropriately. But there is some derogation for diagnostic labs that we need to work at BSL enhanced level, BSL2 enhanced laboratory, but this needs to be appropriately risk assessed within the country. But work still needs to be completed within a sort of microbiological safety cabinet class 2. And this is to protect the worker, because obviously if we're performing manipulations in a laboratory with any liquids, there's likely to be aerosols or droplet contamination of surfaces or the hands and arms of the worker. Obviously, as I mentioned, within a research lab, these using higher volume and higher concentrations, these manipulations are likely to be more risky or hazardous. So we need to make sure this is undertaken within a biosafety level 3 laboratory within a safety cabinet as well. And what we need to do within the research or both laboratories, we need to make sure that our disinfection methods are appropriate and to inactivate high concentrations of liquid cultures within contaminating media as well. So we need to make sure there's validation data there to ensure that our disinfectants are appropriate either for liquid culture and activation or surface decontamination and after spillage or at the end of the day or end of the piece of work. What we need to do also is actually use disinfection at source, which means that we're disinfecting after each step. So we're trying to reduce that hazard from that agent at each step. So if we can inactivate or the agent before it leaves the safety cabinet, that means then we're going to increase our disinfection through autoclaving or incineration after that waste leaves the laboratory as well. And obviously with the diagnostic laboratories, we should be inactivating our waste within the, or inactivating the samples within the microbial safety cabinet prior to it leaving that safety cabinet, especially if we're handling samples at BSL2, biosafety level 2. Then there aren't the processes as there would be for the biosafety level 3. So we need to protect our workers in that environment. So laboratory wastes, again, as the guidance from the CDC and sort of the government and the WHO, we should really be handling, if we're handling agents and infectious agents in a biosafety level 2 or 3 laboratory, we should already have waste streams identified, and we can use this waste, SARS-CoV-2 waste, as through those waste streams as well. So really the waste should be stored within the containment laboratory, and it should only be moved to the autoclave if that's outside of the lab when it's available. And obviously if we can inactivate the waste within the laboratory prior to removal and then disposal, then that makes it extra safe, because we're already handling, we're already set up to handle infectious material within that biosafety laboratory. But also it's identified that incineration is also an appropriate mechanism for inactivation of laboratory waste. But again, such as the healthcare, we need to make sure that we have appropriate packaging and transports are appropriate for these samples, or for these wastes, sorry. So sort of moving on to a slightly different area, as Henk mentioned, it's not just sort of healthcare and laboratories which generate waste. There's waste that's actually generated within the sort of the general population. Oh, is that a question, or is that okay? So looking at residential waste, now for those not self-isolating, so obviously those people that can just go about their normal life, there's normal separation of waste into sort of recycling, and non-recyclable should continue, but there is likely to be an increase in either plastics or non-recyclable, so things like PPE or RPE, so face masks are likely to increase the waste within domestic use, or it's likely to increase during domestic use and can increase the waste levels. But there's also likely an increase in chemicals, so it's either the liquid chemicals which are going to go into the sewage system, or liquid chemicals which might be inappropriately disposed of, which could cause environmental issues, but also in terms of solid, so if they're associated with wipes or absorbent material which might be used to sort of clean surfaces or people's hands. So we need to sort of be wary about that in terms of the waste management. But there also might be disruption in the municipal or private waste services. So that might be due to infections in the operatives, or actually through lockdown of an area, which is seen in sort of the UK. But in terms of those self-isolating within households, again, the waste, and this is taken from sort of the UK guidance, waste is actually still disposed of in the normal waste stream, so sort of municipal waste. But there shouldn't be any recycling of waste in the domestic environment where people have either self-isolating because they're awaiting results, or they have symptoms, or they have a confirmed test, so they actually are confirmed with COVID-19. But the waste should be double bagged, and we shouldn't actually, or those bags should be sealed before they're 70% full. So we're trying to make sure that there's not too much waste in each bag, and it's not likely to split, and the double bagging will actually help protect that during the moving and handling of that waste. And importantly, the waste should be stored within that household for at least 72 hours prior to collection. Now this, as I've shown before, the levels on surfaces are actually inactivated, or should be inactivated within 48 hours. So this actually just builds in a little bit of conservativeness to provide those people handling that waste with a level of reducing that risk from any contamination in the exterior of those bags before they're put out and taken to sort of or handled by the waste operatives. They need to be kept secure from pests, so we're obviously not we're making sure that there's no access to those bags from pests such as things like rats and mice, or even foxes, larger animals, larger predators. But also we need to make sure they're kept safe from scavengers. So people that might look for recyclables to sell, or even for PPE, which has been seen in some countries where people have scavenged on municipal dumps where they've collected RPE, so face masks, to sell on. And in lines with the guidance from WHO, if your municipal services are not available, then safe burying and controlled burning can be completed. But again, this needs to be risk assessed and identified what's the most appropriate options in that situation. And for laundry, should wait at least 72 hours after the last symptom, so after the self-isolation has finished, before taking that laundry to a laundrette or dry cleaners. Because obviously, again, we want to make sure we're reducing the risk from any contamination that might be on those surfaces. Now, so some other areas where waste is generated. So there's schools, workplaces, and actually tourism as well. So obviously hotels and restaurants. And it's likely that extra waste will be generated. And a lot of this, again, will be PPE, but also some lateral flow devices, which I'll come on to in a minute, for sort of point testing to identify if people have any contamination or any SARS-CoV-2 antigens inside. And basically, if we're looking at the PPE, it should only really be handled by the person who's worn the PPE. So we're trying to limit whoever's going to handle that PPE. And for the LFDs, it should be handled by the person undertaking tests. So sometimes including schools or workplaces, if the LFDs are required before entry, then one person is generally doing that testing, and they'll be the person handling those. So it's just be one person, and then they can sort the waste out. And after handling any of the waste, hands should be washed and disinfected, either washed with detergents and water or disinfected using an alcohol hand gel. But again, the waste can be disposed of through the normal route. So there might be an increase in waste. So obviously we can increase the number, or there could be an increase in the number of bins within that environment or that setting to cope with increased in waste. But PPE can be taken home to dealt with at home during household waste. But the PPE and LFD should not be recycled. But because these areas should not have any people who have symptoms or are testing positive, then there's no requirement for them to store them before disposal. So lateral flow devices, this is one I took this morning, so it's quite a up-to-date picture. Now, if we're doing lots of LFDs or in a certain area, as I mentioned before, then the outer packaging can be removed, and that can be recycled prior to taking it into that area, because there's a lot of cardboard waste there, or there might be some plastic waste as well. Or they can be just disposed of through the existing waste streams. But once used, the lateral flow devices and swabs should not be put into waste streams, because obviously they have into recycling streams, sorry, so they can be classed as sort of offensive waste. If they're in a healthcare setting or just in a domestic setting, they can be put into normal domestic waste. So this would be sort of black bags, and needs to be discussed with your waste contractor if they have any issues. So some of the issues from improper waste handling, obviously improper waste handling can present an infection risk. So if we have overflowing bags of waste or leaking bags, or waste which isn't inactivated prior to going to landfill, then there is a risk to people handling, but also people downstream as well, where waste might contaminate the environment as well. We need to sort of keep an eye out for dumping of illegal dumping of waste as well. So we need to make sure that our landfills are sort of used correctly. And in terms of that, we need to make sure that landfills aren't scavenged for recyclable or masks. And also have to, if there is an open burning practice, then we need to identify if there's going to be any chemical pollution from that process. And really environmental issues such as discarded PPE, which we see quite a lot in the UK. People just throw the masks away, and that actually might go to just lay on the side of a road or be contacted by someone else. But also chemicals used in disinfection and decontamination processes entering the normal sort of sewage pathways or potentially contaminating other environments as well. So really it's the end of the presentation. So just a few conclusive points. SARS-CoV-2 is not going to remain viable on surfaces for long periods of time at the levels found in the environment. And transmission by fomites is reduced risk within the environment. But it still doesn't mean that we shouldn't be wearing RP and PPE, as this can reduce the risk further, along with hand hygiene. And we need to make sure that waste is managed through existing waste streams, but we can actually reduce the volume of waste for those more intensive treatments by separating waste at source, to make sure that only the infectious waste will actually go to those intensive treatment management options. So we have a couple of final slides just with reference sources. And these will be sent around, obviously, within the slide pack to you as well. So you'll be able to look at these and click on these and go to the sort of waste management websites as well. So thank you for your attention.